Welcome, good afternoon, everyone. My name is Tímea Gerzsenyi, and I'm a PhD student at the University of Miskolc, Hungary. And today I would like to talk about a part of my PhD research, which is the DNA isolation efficiency of in-house synthesized magnetic nanoparticles. So, as we have heard mainly in yesterday's presentations, magnetic nanoparticles or nanotechnology has a very wide variety of applications. And just to show you a few, they can be used as catalysts, they can be used in sewage treatment, MRI contrast agents, in cancer treatment. But for my research, the most important application is that they can be used for the separation of different macromolecules, like nucleic acids, for example. And so my work, my PhD work is to develop magnetic nanoparticle-based nucleic isolation techniques with the help of these magnetic nanoparticles that are synthesized by our nanoscientist colleagues at our institute. So it would be a reasonable question to ask me, why do we need nucleic acid isolation techniques if we already have efficient ones? For example, there is the traditional conventional nucleic acid isolation, which is a very long procedure, to be honest, and it requires toxic chemicals, for example, phenol and chloroform. But it is inexpensive. There are commercially available isolation kits, for example, affinity column-based, mainly silica column-based isolation kits that are quick to perform. They don't require toxic reagents, but are very expensive. There are some magnetic nanoparticle-based isolation kits as well, and I can say the same for those. They are very expensive, however quick and without toxic reagents. So our protocols that we have developed with the magnetic nanoparticles are quick. They don't require phenol or chloroform, and they are very cost effective. But we wanted to develop a protocol that is at least as efficient as those that are already available on the market. So here is just to show you that our protocols that we have developed are really cost effective. So we developed 3 protocols for the isolation of plasmid DNA from bacterial cells. And you can see the price of 96 isolations here. Double the price, and that's for the price of the traditional isolation. And if we talk about those that are commercially available isolation kits, the price is very expensive. So at first, I targeted the isolation of plasmid DNA from bacterial cells, from E. coli bacterial cells, and I used these 6 magnetic nanoparticles: manganese, magnesium, and nickel ferrite, and their amine functionalized forms. So using these nanoparticles, we described for the first time in literature that they can be used for DNA isolation from bacterial cells. And here you can see the brief methodology, the steps of the isolation. So first we lyse the cells, we create a high pH for the denaturation of the macromolecules to precipitate everything, and then we renaturate only the plasmid DNA, the small circular covalently closed plasmid DNA can be renaturated by neutralizing the pH. Then we add the magnetic nanoparticles that are in a binding buffer, dispersed in a binding buffer. And given the fact that the nanoparticles, the surface of the nanoparticles is positively charged and the DNA has an overall negative charge due to its phosphate backbone, there is an electrostatic interaction between them. So when the DNA MMP complexes are created, we use a strong external magnetic field to separate these complexes, and then we can change the supernatant, we can remove the contaminants. And at the end, we just create an environment that supports desorption of the DNA from the surface of the nanoparticles. And then we just get the pure DNA. So we have developed 3 protocols for the isolation of pDNA, and the optimization of the protocols, because we have tried several types, several protocols, and the optimization meant to change the adsorption environment, so the binding buffer and the elution buffer, which was the desorption environment. One main component would be the sodium chloride, I would say, that is present in the binding buffer of protocols number 1 and 2, and the elution buffer of protocol 3. So if we put the DNA in a low salt concentration buffer or water, there will be an electrostatic repulsion between the negative charges of the DNA, of the phosphate backbone. So the DNA will have kind of an elongated conformation. And this will result in even one DNA molecule covering almost all of the binding sites of one nanoparticle. But if we put the DNA in a higher salt concentration buffer, then there is a counterion introduced to the buffer, so the negative charges will be neutralized, and then the DNA will have a more compact globular conformation, so more DNA can be loaded on the surface of the nanoparticles. So all in all, we can say that with protocol number 1 and 2, we can get more DNA from the bacterial cells and less with protocol number 3, but still all 3 are good, are yielding good purity DNA. So I evaluated the extracted DNA. I checked if it can be used in further applications, in downstream applications. First, of course, I checked the concentration, I checked the purity of it, and I set this threshold 10 microgram per milliliter, because this is the highest, the maximum extraction capacity of most magnetic nanoparticle-based isolations if we consider 5 milliliter cell suspension, if we extract from 5 milliliter bacterial cell suspension. I did agarose gel electrophoresis to see if I have clear DNA bands. And then I checked if the extracted plasmid DNA can be a good template of several enzymes, like restriction endonucleases or qPCR, so polymerase enzymes. So I had 3 protocols and 6 magnetic nanoparticles, and I did all of these, I tested all of these combinations at least 3 times each, and this is the summary of the isolation efficiency. So there is at least 1 protocol in each case which was suitable, which checked all the 5 criteria of the evaluation. And as you can see, the magnesium, the amine functionalized magnesium ferrite checked the criteria for all 3 protocols. So I consider this the best magnetic nanoparticle, I would say the less sensitive to different environments and buffers. So given this, I continued my work to try to isolate not just plasmid DNA, but genomic DNA also with the best performing magnetic nanoparticle. And I used gram-negative and gram-positive cells, so 4 types of bacterial cells for this. Again, there's the methodology of the isolation. So after lysing the cells, we add the nanoparticles. This is the same composition of the binding buffer as in protocols number 1 and 2 in case of plasmid DNA isolation. For the washing steps, I used different dilutions of ethanol, and on a high pH, I did the desorption of the DNA from the surface of the nanoparticles. Here you can see the agarose gel electrophoresis picture of the different genomic DNAs isolated with the magnesium ferrite and the concentration, which is the genomic DNA extracted with the magnesium ferrite nanoparticles can be compared to those that were extracted with commercially available isolation kits. And those that were extracted from the gram-negative cells had even higher concentrations. So I would say that these are good results, again, in this case. I performed qPCR with this program, and I used bacteria-specific primers. So here you can see the CQ values of the qPCR for each bacterial cell. So the values are between 20 and 25. Everything which is above 30 should be considered as an artifact. So I have these 2 negative controls, the non-template control, which has a high CQ value and an intensity because of the primer dimers present in the reaction, and this is the non-primer containing control. So these are also good results, and we wanted to examine the extracted DNA further, and as the plasmid DNA that I've extracted from E. coli contains ampicillin resistance gene, I transformed this so-called PBAC24 plasmid DNA to competent E. coli cells, and I cultured them on ampicillin containing LB medium. And you can see that all of the plasmid DNA got transformed because I saw these nice colonies, E. coli colonies, that grew on the LB medium. And one last thing that we did for the evaluation of the extracted DNA, actually this was a request from one of our reviewers, to see if we can extract the DNA with the magnetic nanoparticles from a more complex sample. And so I mixed blood serum with bacterial cells, and I did the same isolation protocol that I've described earlier, and it worked pretty well. So this is the concentration that I've got. I had 2 elutions. So actually, I also tested this, that if I added additional elution steps at the end of the isolation, do I get more DNA? Is there any more DNA remained or adsorbed on the surface of the nanoparticles? So I did test this, and I went up to 4 elution steps, but I realized that I only need to do 2 because, as you can see, at first I got around 300 microgram per milliliter concentration, and at the second one around 100. So this is it is worth to do a second elution. Also, the purity of the DNA was in the accepted range. So we have proved that the isolated DNA can work, and it can be used in downstream applications. So we just published an article about these results, and our further goals are to broaden the application of the nanoparticles. For example, to isolate from a more complex sample, to isolate from soil sample or to wastewater, and also to do RNA isolation, which will be a little bit more complicated given the fact that it's a more unstable molecule than DNA. But we have started these tests as well, and it looks like it's gonna work. So that would be my presentation, and I'm up to questions if you have any, and thank you for your attention.